Alright, did you guys grab, I need you guys to grab one of these. I'll put it to the side and do it later. Okay, I know you guys have been listening to the Betty Wap, but I'm going to do the Fantastic because it's a little bit longer, so you have some more time.
Welcome, I'm so happy you came by. For a lesson about the essence of the Iowa living life. DNA is a topic that's so fantastic. We're talking about the oxyribonucleic acid. If you've ever wondered about development in elephants or bumblebees, talking juice, well, DNA is the recipe. Eat its home in a chromosome in the nucleus. DNA doesn't us genetically controlling us. Frozen words, infectious germs, mommy's egg and daddy's sperm. DNA is the information linking up the generations. It's a code of life, so listen up well. Because DNA is the molecule in charge of every cell. DNA, fantastic, deoxyribonucleic acid. We're talking about the deoxyribonucleic acid. DNA, fantastic. The shape is key, probably well known to you. Double helix, twisted ladder, or a double corkscrew. Imagine a twisted length of tape with deoxyribo sugars on the edges with phosphate. Sugar phosphate, making up the sides of the ladder with nitrogenous bases on the inside. With the bases like the bonds together with deoxyribo sugar and a phosphate make a nucleotide. Deoxyribonucleotides are the monomers.
I'll tell you guys next week.
DNA polymer, 0.34 nanometers. Marks the space between one nucleotide and the next one's place. Before the helix to make one turn takes 10 bases in a row.
This is in your nose,
Since
so don't
space
sit there
between
with the blank
nucleotides
stare.
is 0.34, one turn takes nanometers, 3.4. So stylish, so slick, first described in 53 by Watson and Crick. And don't forget Franklin, she took that famous X-ray pic, but she died, so she never got a Nobel Prize. Fantastic deoxyribonucleic acid. We're talking about deoxyribonucleic acid. DNA. Fantastic. So nitrogenous bases come in one of four varieties, known by the letters A, T, C, and G. A is for adenine, thine's got the T. C, it stands for cytosine, guanine's got the G. A and G with 2 nitrogen rings of purine. C and T with 1 ring are pyrimidine. C only bonds with G, A only with T, because like puzzle pieces, they're complementary. They fit together, snuggled up like hand and glove, like enzymes and substrates, like people in love. A, T, C, G, now shaped like lock and T. Hydrogen bonds, either 2 or 3. Hydrogen bonds, you know they aren't very short, but they're enough to hold DNA together all day long. But when
Actually, no, that's silly. Don't skip your over. Be careful with the
DNA with the 2 nanometers. DNA's double-stranded, looks pretty swell. The strand orientation is anti-parallel. With one strand standing up, the other on its head. It's hard to get together, quick as Watson said. DNA, fantastic deoxyribonucleic acid. We're talking about the deoxyribonucleic acid. DNA. Fantastic. Sugars and phosphates make the backbone, the structure, but the sequence of bases is where you find instructions for development of bodies in the cytoplasmicity that constitutes like miracle and coats and golden algae. The sequence of bases is unique, it's a special batch. Unless you're an identical twin, it's nowhere matched. And you can bet, and if you're not Django Fett, that you've got no clone. No one like you's been known. Our basis comprise our individuality. You might have an A where I have a G. Explain it why I'm bald and you got all that hair. These differences can show up anywhere. The protein hemoglobin, listen up well, it carries oxygen in your red blood cells. Hemoglobin is made of 4 protein pieces, a trait widely shared among animal species. Hundreds of bases spell what hemoglobin piece. Hundreds of A's, G's, T's, and C's. The gene starts CACGTGCAC, then TGAGGACTCCTC. The key is these bases are information for hemoglobin's function and conformation. Hundreds of bases in a predetermined order. A single change brings on a major disorder. Change T to A in one single spot. This little one mutation might not seem like a lot. Binding to adenine might not seem that big to you, but baby, that's the cause of sickle cell anemia. DNA.
That's what we're talking about today.
Fantastic deoxyribonucleic acid. We're talking about the deoxyribonucleic acid. DNA.
Fantastic. Yes, sir.
Are we gonna do the alien thing?
Oh, the what?
The alien.
No, the alien, yeah, first things, that's homework. I'm taking that up by Friday because we have to talk about gene mutation today. So I had to harp on first period really hard. Don't get out and start working on those because we're learning new material today, all right, on gene mutation. But yesterday you guys got to experience start and stop codon. So what is a start and stop codon, Carly? what is actually being coded. When you did your alien stuff yesterday, what were you actually coding? No, well, be more specific. It's located in the gene. Yeah, kind of. Colin?
I was going to say
DNA, what do you mean DNA?
like they were just trying to find out if you're translating the DNA.
What do you translate the DNA to?
RNA.
RNA, which then translates to what? Your amino acid. Now, do you guys remember what the start codon was for the amino acid? No. Oh. No, it's not mRNA. mRNA. What is mRNA? Oh, no. That's the next step. That's just regular RNA. Naomi. MET, yes, that is your amino acid. That's your start. Everything about you that starts. All right. I'm going to get more in depth into that because now that I talked to you, I can tell I'm going to have to stop the video like halfway through. It's going to make sense, okay? So go ahead and put that away because it affects your amino acids. Now, I want you to clear everything off your desk. The way I'm teaching this lesson is we're breaking it up into 3 different parts. So everybody look back at our learning target. In the back. We have 2 things that we're trying to learn from this lesson. 1, I can explain how a mutation occurs, and 2, I can describe if a mutation would be beneficial, neutral, or harmful for an organism. Okay, that's it. That's your 2 things. Now, if you look on that board over there, I'm not making you take notes over this. It's only 6 minutes long, and it's over our goobery, like our goofy people, okay? But I am going to ask you guys as a group to help answer those questions. Those are kind of like higher order thinking questions. So read those, be thinking about it, because then I'm going to be randomly calling, and we're going to have like a little class discussion. And then you have your activity that you picked up when you walked in. So there's 3 different ways that we're trying to learn this, because this is new information. You're going to try to get information from the video. We're going to have a class discussion, group discussion, and try to ask questions where you can use me to help answer them. And then you have that, which that gives you information, and you can design a foldable where it helps you create a graphic organizer. I went blank. So what?
Whenever you said about the...
You look at your napping. Sit up, please.
Are we gonna like answer as a group?
Like we're gonna watch it, and then I'm just gonna randomly call someone out, and then just pick. It's going. I mean, I'm hoping that you guys kind of take over and you ask the questions that you want to ask, because gene mutation. I know we kind of jumped in this a day earlier than the other classes. This stuff's like really, I don't want to say cool, but I'll say it's very interesting. I want to show you some pictures of some actual real life gene mutations, and they're really, really interesting. So I would really prefer you not to move your desk around, but I need you to kind of turn around, face the front. If you guys aren't paying attention, you know you're going to get a warning and stuff, because you know questions are coming. So Deb, up here. It's 6 minutes long. Hit the lights, please, whoever. Now you're going to have to listen to this loud. Oh! You're not missing anything. It just says biology.
DNA mutations.
Mutations are changes in the DNA sequence.
Mutations. DNA getting messed up, leading to all sorts of problems. Or to something.
All right, mutations. Let's talk about it. Remember our 4 bases in the DNA strand, A, T, C, and G? Well, a mutation occurs when one of these letters gets changed to another letter.
Before we go on, let's take a second to get something straight. The truth is, it's more correct to say that a mutation occurs from a changed nucleotide rather than a changed base. Remember that a nucleotide is simply a unit of DNA or RNA that includes a base, along with the sugar and phosphate group that form the backbone of DNA. Don't worry about these specifics too much right now. Just be aware of them.
You may be wondering, how does this happen?
Well, mutations are caused when one of the nitrogenous bases gets messed with.
Ouch. That can't be good.
So what's the effect of little base change in the DNA? Well, let's consult the central dogma. Remember, the central dogma is the process by which an original DNA code is transcribed into mRNA, which is then translated into protein.
Remember, each protein is made by a sequence or sentence of codons. Each codon, like a word made of 3 bases or letters, refers to a specific amino acid, and a protein is a sequence of amino acids. Here's a normal amino acid sequence.
And I'm stopping it because of the way you guys were answering the bell ringer, okay? Because how many of you feel like remember you were creating the aliens yesterday, how well you were doing? Like, I need to at least know, if I gave you a DNA strand, so give me a thumbs up, a side, and a thumbs down, and be honest, I get so frustrated when I see someone that I just know doesn't know how to do something, like I've watched them for 10 minutes, and then they give me a thumbs up, like I got it, be honest. If I gave you a DNA strand, would you be able to convert that to an amino acid? Give me a thumbs up if you can do it, give me a sideways or a thumbs down. Okay, so I have, I'll get my 2 first thumbs down. Which one? I need to know, because I'm writing it. Okay. Okay, you can put it down. So look at this up here, so I'm stopping it. How is this, just this image, similar to what you guys did yesterday? I'm doing random stuff, so you might as well not even raise your hand today. Josiah, your hand is up.
It's kind of like we did yesterday because it's the same answers. Like, we got the same exact thing up there.
Okay, yeah, you're getting the same answers. Can you explain to me what is actually happening? Like, if I asked you guys what goes with T,
It goes
what would you say, everybody?
A.
A, right? You all have that. Every one of you said it, okay? You can decipher the code. But, you okay, Colin?
Yeah.
You got a headache?
My braces are hurting.
Okay, alright. But, I'm trying to get you guys to see the big picture here, okay? Just because you can look in a letter and say, oh, A goes with T, what's actually going on? What are you moving from? What are we starting with?
Find me.
Hold on. Let me just do random. Blake, what would this first line represent? Gene 1. I'm looking for something a little bit more. Notice that hey, I'll give you a little hint. Notice that it has a T in it.
A.
No. Notice that it has a T. Where are we getting this information?
The thingy, wherever that you
The
chart
thingy?
it with.
The thingy. The thingy. What's the thingy?
It's the thing that you chart it with.
Huh? What did you start with? Exactly. What do you start with? What is this line? You're gonna be so mad
The mutation.
when you figure out. No, we haven't even talked about the mutation yet. What does this first line represent? It's not that hard. You did it yesterday. I watched you. You did it well. What did you start out with? Where is this information coming from? Oh, she's not in here. Damn.
DNA.
DNA, okay? That's why I said it has a T, because thymine is in DNA, but not in RNA. So Blake, this line, that's your DNA. Then it goes to what? Adriana?
RNA.
RNA, which then goes to what, Bree?
Proteins.
Well,
Well,
what is a protein? What is a protein? This right here, listen, Bree, that's not that bad of a guess. That's actually a really good guess. But is this a protein?
amino acids.
It's an amino acid. So a protein is what?
RNA.
It's a what?
RNA.
Oh yeah, a bunch. It's a sequence. What does sequence mean, Hannah?
In order?
Yes, in order. It's just a bunch of amino acids in order. That's all protein is. So all of these are your proteins. Before we do mutation, you have to know that, which you do. I'm just trying to get you guys to see the big picture. All right, I don't want you guys to be robots and just know that goes with that. Like this is why. All right, you get your DNA to RNA, which gives you amino acids. When you get them in order, it's your protein. All right, does that make sense?
Yeah.
A little bit. Now, we're going to do what? I'm sorry, sweetie.
Was sequence really right?
Yes, in order. A sequence. Like you have a sequence every day. You go to first period, second period, third period. It's the same way. There's a certain sequence in your DNA and RNA that goes in order for your amino acids that creates protein. Okay? Now, here's where you get your new information. Which, honestly, I think you guys get that okay. I just wanted to make sure, so this part would be a little easier. Alright, this is what mutation is.
The first line shows the DNA sequence, and the next line shows the transcribed RNA. The third line is the amino acid sequence resulting from translation of the RNA. Normal, normal, normal. Now, let's see what happens when the DNA gets mutated. Now, let's say this cytosine here, C, gets changed to an adenine, A. Considering how precise those bases are about who they get paired up with, you know, A with U and C with G, this could get interesting. Let's see what effect this has on the protein. First, watch what happens in transcription. In this case, the imposter A gets transcribed into a U. So, our mRNA codon becomes UCU, which translates to serine, not alanine. This type of mutation is called a missense mutation.
Okay, that's the first one. Does that make sense? Can you see how one little bitty change changes that to here, and it completely changes the amino acid? Why are you guys yawning so much? You're all like really tired. Okay, it's changing. Get it out, Deb. It'll be worth it, I promise.
Because in that mutant codon, there is a mistake protein being translated.
Actually see
So a missense mutation is when just one base has been changed, resulting in a codon for a different amino acid. Well, at least the codon is referring to an amino acid, so it still makes sense. It's just the wrong amino acid.
Like if I said the sky is purple. That would be wrong, but it would still be a complete sentence, and purple is still a color. It's just the wrong information.
Missense mutations can be the cause for certain diseases, such as sickle cell disease.
There's another type of mutation, a nonsense mutation. This is when the codon for the amino acid gets changed into a stop codon, which causes the translation to get stopped before it's been completed. Like if I said, the sky is, and then just stopped. That makes no sense. Get it? Nonsense mutations can cause genetic disorders.
A terrible, terrible situation. How long have I been saying? Grammar, 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 punctuation, proper spelling. How else do you expect to communicate properly in this world? What are we talking about?
Now let's look at another type of mutation. This one,
It's so
called a
tough.
frame shift mutation, gets pretty messy. Watch this. Say we insert an extra thymine in the DNA here. What effect will this have on the outcome of the translation process?
Well, all of the codons are always grouped
This
as
is where you get your
nucleotides
really, really
each,
bad
right?
ones.
When you throw in an extra nucleotide or letter, it's going to get grouped into a triplet, pushing one of the letters from that triplet into the next, pushing one of the letters from that triplet into the next, and so on, jumbling up all the triplets.
Say I had the sentence, fat cat ate mud, and for some reason, I had a computer that only allowed 3 letters per word. If I added an S to the end of fat, the sentence wouldn't just read, fats cats ate mud. Instead, the computer would focus on having 3-letter words and changing it to fat, ska, tat, emu, d. Complete gibberish. You can see how disastrous frame shift mutations can be.
They can also occur when a nucleotide gets deleted, and things end up getting just as confused, leading to missense and nonsense translations all across the board.
One example of a disease caused by a frame shift mutation
That
is
one's
Tay
awful.
Sachs.
Finally, we
Hey,
have
Sam,
something
it's on- I
called
wrote
a
it on the board.
synonymous substitution. This is sometimes called a silent mutation because although a base in the DNA gets changed, this change has no effect on the final protein.
Think about it. Remember how there are multiple codons that translate to the same amino acid? You could say they're like synonyms, 2 different words that have the same definition. So, when a mutation causes for a synonymous codon, it translates to the same amino acid as it would have in the first place. So, these synonymous codons are sort of like a built-in safeguard against the disastrous effects of mutation. Neat!
That's all for now, folks.